Hi, my name is Jacqueline, and so this is my group's gut microbiome lab. And for introduction, a gut microbiome is generally just the different types of bacteria in your gut, and it can be related to the health in your body. So there's two different physiological conditions that we stated that could be involved with the gut microbiome, and they are type 2 diabetes and autism. So type 2 diabetes, it causes an imbalance in blood glucose levels, so that the gut microbiome will reshape the intestinal barrier functions and host metabolic and signaling pathways. This can directly or indirectly cause a rapid progression of insulin resistance, which leads to your type 2 diabetes. And there are thousands of different microbes in the gut, and with certain dietary factors, it can strongly affect the gut microbiome. So therefore, type 2 diabetes people's lifestyles could highly affect their gut microbiome. For autism, these two are related because of the brain-gut link. A study was actually found on rats that did not show signs of autism, but when they were introduced to feces from humans with autism, they responded showing signs of autism. So while this does not prove that bacteria in the gut can cause autism, it does show that the makeup of the gut can contribute to the hallmark features of this disorder. The way this changes behavior is since the brain and gut are connected, it changes the way the brain makes these different proteins. But the relationship is correlative. For both these, they are correlative. So our first question asked about the health effect on the gut microbes of dogs. So our hypothesis was if the dog's healthier, then there would be more bacteria present in the gut microbiome. Our second question we asked is if the age would have an effect on the gut microbiomes of dogs. So then our hypothesis ended up being if the dog is younger, then there would be more bacteria present in the gut microbiome. Okay, so for our materials and methods, we had round one of animals compared. We had 4 samples. The first 2 were poor health, and they were a 12-year-old female border collie and a 9-year-old male beagle. And then our other 2 were our good health dogs, and they were a 6-year-old male shepherd hound mix and a 12-year-old female West Highland Terrier. So for this experiment, we did 3 steps. The first one was purification of DNA from the fecal matter using the QIAGEN kit. And we did this according to the manufacturer's instructions, so we didn't change the protocol at all. Second was quantification of the DNA in samples. And the third was qPCR setup and amplification of the DNA. So using the quantification of DNA via nanodrop spectrophotometer, we use this DNA to absorb light. It absorbs light at 260 nanometers, and the protein would absorb the light at 280 nanometers. For normalization of DNA samples using the qPCR, we used primers, and these are forward and reverse primers which bind to DNA and allow for DNA polymerase to bind and begin synthesizing DNA. So this was the bacterial and fungal primers. And then we use cyber green, which this binds to the double-stranded DNA, not the single strand, and emits fluorescent light, which correlates with amplified DNA. Thus, that's our initial samples amounts. We also had to do second and third round of animals compared. So we had another 4 samples. This was the where we compared age. So we had a 4-year-old female pit bull and a 5-year-old male pug. Then we had a 10-year-old female boxer and an 8-year-old female boxer. So we chose these samples from other groups because of their CQ values. Round 1 samples produced CQ values over 25, which this indicated absence of the bacterial DNA. Or it could have done contamination throughout our experiment, but instead we decided to switch them anyways. And then we did not perform second DNA purification of the quantification of the nanodrop. We only performed additional qPCR measurements for the phylum of the bacteria. So now our new question turned into, does the age of dogs affect the gut microbiome? And we used the primers for the phyla for the second round were the Acetobacteria and the Firmicutes, and then for the third round, the phyla were Actinobacteria and Bacteroidetes. Okay, so these are results for our experiment. This is for the UV spectrophotometry of our first round. So we measured the absorbance, the ratio, and the concentration. So the absorbance was 0.95, 0.042, and 0.082, and 0.105, which are all good numbers for these 4 samples. And then we had ratio, which would measure the purity, so the absence of like contaminants or like RNA. So we had 1.43, 2, 2.69, and 1.66. We wanted between 1.5 and 2. So 3 of these were good enough, but we had 1 on the outs. And then concentration, we had 47.5, 2.1, 4.1, and 5.2. So the concentration would tell us if we had enough to be diluted, and all of these worked out. So then we moved on to the qPCR, and we did a positive control and a negative control for the qPCR. So our positive control showed a good amount of DNA present due to it being their CQ value, which this should be under 25, because DNA would be present. If it was over, that means there's no DNA present. So we had it at 13.43, which is good. Our negative control was over 25, which made sense because we didn't put any DNA in, so there shouldn't be any present. But for our 4 samples, we had a few that were over. We had 1 that was under 25, but then we had 3 that were over. So therefore, there was no DNA really present. And this is for bacteria and fungi. So then we moved on to our second round, and we changed the samples, and we did the phylum. So we did Acetobacteria and Firmicutes on this, and their CQ values for Acetobacteria were well over 25, so there was no DNA present. And then they were at 32.92, 31.49, 30.7, and 29.73. And then for Firmicutes, they were much better, or they were a little bit better. They had 3 under 25 at 24.46, 23.37, 24.03, and then there was 1 that was 25.02, but that's still a lot better than Acetobacteria. We decided to still change it. We did 2 different ones in the round 3. So we did Actinobacteria and Bacteroidetes. So Actinobacteria had all numbers over 25, so no bacteria or no DNA was present, which that's not what we really wanted. And then we had Bacteroidetes, again, all numbers over 25. So out of the phyla, the Firmicutes worked best for us, but we decided to just test those just in case. So, yeah. Okay, so for our discussion, as we said, extracting the DNA involved measuring the absorbance, the ratio, and the concentration. So the absorbance at 260 nanometers. A concentration greater than 1 identified high enough of concentration to dilute to the solutions. So all concentrations recorded were greater than 1, which is what we wanted. The ratio helped identify the purity among the DNA, which absence of RNA. And the samples 458 and 318 in the first round had ratios between 1.5 and 2, therefore a greater purity among the 4 samples and less RNA was present. The negative control in the first round had a bacterial CQ of 27.67 and a fungi CQ of 34.08. So these numbers showed there was no DNA to be found, which is accurate due to no addition of the extracted DNA or the control DNA. The positive control in the first round of the qPCR had a bacterial CQ of 13.43 and a fungi CQ of 31.71. Both of these numbers should have been under 25 due to the addition of the control DNA, but and knowing the exact value that should be present, we did not conduct positive controls for round 2 and round 3. The negative control in round 2 and 3 of qPCR were values well over 25, so I stated before that's what we wanted, and so that worked out. The differences among the gut microbiomes did not vary as much as we would have liked. Our 2 younger dogs had bacteria present in their microbiomes like we expected, but so did one of the older dogs. So, and it's because it had CQ values of 24.46 and 23.37, whereas the older dogs had values of 24.03 and 25.02. So that 25.02, definitely didn't work out. With that being said, our results did not support our hypothesis that our dogs, being younger, that will have more bacteria present in the gut microbiome.